So hopefully by now you've read through the chapter on the forces of evolution, talking about what is evolution, and by forces we mean what causes evolution. How does evolution happen? Well, to sum it all up, the forces, with our understanding today, the modern synthesis of evolution, what causes evolution can be considered mutation, migration or gene flow, genetic drift, and of course Darwin's natural selection. Darwin had proposed that evolution was the result of evolution and adaptation, actually, was the result of natural selection. But in the 1940s, there was a melding of population genetics and classic Darwinianism that acknowledged the importance of mutation, gene flow, and genetic drift in evolution. We'll look at each force or mechanism in detail. But while we're looking at the forces, keep in mind that mutation and recombination generate and increase diversity within a population. And really, mutation is the only way that you can get brand new variation. Genetic drift and natural selection reduce diversity. They eliminate or remove diversity from the gene pool. And by gene pool, what do I mean? Well, the gene pool is that collection of alleles or that collection of variants or traits within a discrete population. What is microevolution versus macroevolution? Microevolution is simply change in the frequency of alleles or change in the frequency of particular traits within a population over each generation. Slight fluctuations, but a new species does not arise. There's not enough change to consider that this population has now diverged into a new species. Macroevolution is where enough differentiation occurs in the allele frequencies or trait frequencies or a trait to consider that, yes, this is a new species. The question arises in evolutionary studies, what is a population? Well, it depends. Depends on what your research question is. The population that you're defining may be simply the population of Portland, or maybe the population of Portland State University students, or possibly it could be comparing two populations of macaca mulata, so two populations of macaques. It depends on what your research question is, so what a population is variable, but must be defined when you're asking your research question. Mutation pressure is the idea that over time and generations, as mutations accumulate in an allele, then the frequency of the original allele decreases. So for example, you have a population that has a gene that 100% of the population has the same allele. Then there's a mutation in a germline cell, and an offspring has this mutation. Now you have a mutation in the original allele, and so the frequency of the original allele has decreased. So again, mutation is really the only way to introduce novel variation into a gene pool or into a population. And of course, that mutation must be in the germline, meaning in the gametes. Otherwise, it's not passed on to the offspring. The force of genetic drift can often be a difficult one to explain and get a handle on, but genetic drift is simply a random stochastic process, almost meaning random luck, that just you have fluctuations in the frequency of alleles or frequency of particular traits in a population simply because of just random happenings. There's no selection. It's not because the trait is favorable or maladaptive, meaning not favorable. It just simply is random luck. Things happen that will impact the frequency of particular alleles. With the force of genetic drift, population size is critical. The smaller the population, the larger the impact of drift. Drift will happen faster, and there'll be a more dramatic change in allele frequencies. So think about, you have a population of 10, and just through random luck, 3 individuals are removed from that population. You've lost 30% of the population, 30% of the variation, 30% of the gene pool, versus you have a population of 1,000 and you lose 3, you've lost 0.3%. Fixation is when an allele reaches 100% in a population. It's the only allele in that population. It's become fixed. So when new alleles arise, over many generations or over time, they may become fixed, or a new allele may be eliminated. We also can have balancing selection, where an allele is maintained in a population. Migration, or gene flow, is the force of evolution where genes are introduced into a population from another population. So the genes or the alleles are flowing from one population into another population. So think about Neanderthals and modern humans. They came into contact as modern humans were expanding out of East Africa, expanding through the Levant and into Europe. These two populations, when they came in contact, mated and shared genes. So new alleles were introduced into modern humans, and new alleles were introduced into the Neanderthal population. Didn't work out so well for the Neanderthal population. Bottlenecks, founder effect, colonization. These are processes that result in a population with reduced genetic diversity, a reduced number of alleles. Bottleneck is where you have a large population, genetically diverse, multiple alleles. Some event happens that eliminates a large number of those individuals fairly rapidly. And so thus you have this winnowing effect. The outcome is a small population with reduced genetic diversity. Founder effect is when you have a small population of individuals that leaves the original parent population. They migrate out of that parent population and they found a new population somewhere else. So they take with them their alleles. And the new population will only have those alleles until new mutations accumulate or until they come into contact with other populations where genes flow into this population. So, founder effect, a smaller population takes with them their alleles, and the new population just has those alleles that were in that founding population. So, natural selection acts to remove individuals from the gene pool. And natural selection results in differential reproductive success. So, what is differential reproductive success? Well, this is simply that some individuals produce more offspring, some individuals produce less offspring. But it isn't simply that the individuals that produce more offspring are the most successful, because if their offspring don't live to adulthood to reproduce themselves, then those parents still have not been successful. They haven't contributed to the gene pool. Selection can be positive, can be negative, and can be stabilizing. So, negative selection is removing an allele or a trait from a population. Positive selection is selecting for or increasing the frequency of that allele or trait. And stabilizing selection or balancing is maintaining multiple alleles within a population or maintaining an allele in a population that seems like it should be negatively selected or selected against. So an example that we'll talk about later in the quarter is sickle cell trait. This mutation that causes sickle cell is actually maintained in the population because the heterozygote, an individual who carries one normal allele and one sickle allele, actually has an advantage in areas where malaria is endemic. So this begs the question, are all traits adaptations? Are all traits under selection? And it turns out that not all traits are actually adaptations. So not all traits, features, behaviors are adaptations. We can see traits or characteristics that are what we call exaptations. These are characteristics that were once an adaptation but are now serving a function that is not related to or different from what the original selective context was. So, what is an adaptation? What's the concise definition of an adaptation? It's a feature, a trait, or a behavior that has been selected for its current function. It increases reproductive success through its current function and was selected for its current function. So what do I mean when I say that males have greater variance than females? This is the idea that there's a wider range in variation in reproductive success in males compared to females, or reproductive opportunities for males. So when you look at most females, and you look at females across different types of different species, different populations, there's a narrower range of reproductive events. yes, we do see the occasional female that has maybe, if we're thinking about human populations today, maybe 20 offspring. But in general, most females tend to have similar numbers of reproductive events, similar opportunities for reproduction, whereas males have a much wider range in the opportunities for reproduction and number of reproductive events. Effective population or effective population size is the size of the population of individuals that are actually reproducing. So you may have a population of 1,000 individuals, but if only 10 of those individuals are actually capable of reproducing and having access to reproductive events, your effective population size is much smaller. One of the most difficult concepts in or definitions in evolutionary biology, evolutionary anthropology, is how do we define a species? How do we delineate species? And the traditional biological concept is that where a species is a population or a group of individuals that are able to mate and produce viable offspring. But of course, the problem there is that we have hybrids. Think about mules. Mules are the offspring of a horse and a donkey. And so certainly horses and donkeys are two separate species, but they're capable of producing offspring. What about fossil species? Later on in the quarter, we're going to be talking about paleoanthropology and looking at the fossil record. We can't tell if fossils could interbreed, so how do we determine species? We can look at time, geographic distance, but we don't know for sure that fossils that are separated by 300,000 years and 10,000 miles could or could not interbreed. So we look at morphology and the characteristics. So tempo and mode. This means the timing of evolution and the big picture of how it progresses. And the classic Darwinian model of evolution is anagenesis. And this is where you have a lineage that's slowly, gradually accumulating changes over time. But this isn't really what the fossil record shows. The fossil record shows long periods of stasis, long periods where there's limited change in a species or a lineage, and then rapid bursts of speciation. So this led Niles Eldredge and Stephen Jay Gould in the 70s to propose a model of evolution called punctuated equilibrium. With punctuated equilibrium, the emphasis is not so much on one lineage slowly, gradually evolving into a new species, but that you have multiple branching events or multiple speciation events where you have branching and diverging of populations. Let's return to the concept of the biological species, or as Ernst Mayr promoted the biological species concept of a population of individuals that are able to breed. Again, this is very problematic with the fossil record because when we recover fossils, fossil individuals, pieces and parts of fossils, we don't know if they were able to interbreed. We're limited to defining our fossil species on morphologic and metric traits. But the problem is when we have two fossils that are separated by maybe 100,000 years, but they're both from East Africa, and we're defining the species based on the morphology, we don't know what the range of variation in that paleospecies would have been. So, for example, with range of variation, Australopithecus afarensis is a paleospecies, a paleo hominin or an extinct hominin, an ancestor in our lineage, that there's a discussion of what is the range of variation um, in size, in canine size, and in overall size of the individuals within this species. There are two sites, Hadar and Laetoli, that are both in East Africa. And a number of Australopithecus afarensis individuals have been recovered from these sites. And the range of variation of size is fairly great. So the discussion has been, well, is this simply male-female sexual dimorphism? Is this just the normal range of variation in the population, or do we actually have two different species? Another way to think about this issue of the range of variation, if we think about the smallest humans on Earth today, so a population of humans that are of small stature compared to a population of humans today who are known for being very tall. If you were to sample simply one individual of the smallest stature, smallest height population, and an individual of the largest height population, and nothing in between, if you were defining the species based on height, then you might say that they're two separate species, when in actuality, they're the same species. They are Homo sapiens. They are both modern humans. We'll go into more detail about Hardy-Weinberg equilibrium in a separate lecture.